Let's move to maybe to David. Yeah.
Well, I would like to echo all of Paula's platitudes and thanks to everybody. And I'm not going to repeat them. I know what it means for folks like you to take time out of your busy lives and come together to do a study and a report like this over the course of a year, and we're grateful for it. I'm David Tomko, and I represent Space Life Sciences. In particular, I'm the program scientist for space biology. Is one of these things a pointer? Probably not. Oh, one here. And, aha. So, Space Life Sciences spans the whole of life systems, all the way from microbes to humans. And we have a little bit different problem than people who work in buildings on Earth. The buildings in space, like the space station, are there. We have to bring our own air, get rid of our carbon dioxide, get rid of our waste, bring our water or make water, and there are all kinds of efforts to try to do a lot of these things. We're managed in kind of a fractionated way. There's a lot of work going on in the basic sciences and the biological systems emphasis, and there's a lot of work going on in the human area. NASA has been in the business of looking after the health of the astronauts for 40 years or 50 years. So there's a lot of work that has preceded the past decade of looking at pathogens and microbes that are specific to humans. We are now in kind of a new era, and that is an era that began with a set of recommendations that were given to us by the National Academy, and we have used these to build programs in space biology, in physical sciences, and in human biology. There was a clarion cry for two things that are relevant for this particular committee and this particular topic. One is a recommendation from this 2011 report that we should use the ISS as a microbial observatory. So we have taken that seriously, and we've, as committees like yours are wont to do, they didn't really totally define what it is that they meant by that. So we have fundamental science, we have applied clinical science, we have epidemiological environmental surveys of what's on there. And we are also charged to look at the evolution of microbial species under the stresses of spaceflight, which are the total containment, the radiation, weightlessness, and there's still argument over small critters and how they're affected by gravity, how they're affected by being in a free fall. I know that some people in this room are engaged in at least one, engaged in similar studies, and one of the things I would like from this committee and this report is a recommendation for us to be better coordinated across all of the folks who are doing stuff like this. I think that that's really important. I'm not going to go over all of these, but obviously in order to do science studies, if you want to put together microbes and mix them and study them or bring them up and bring them back, subject them to radiation and then subject them to spaceflight, you need hardware. And I've got a couple of slides in here that represent some of the hardware capabilities for doing cell culture and incubators on ISS. It's not an easy task, and the studies are few in number because it's difficult. They require upmass to get up there. They require a lot of preparation. This is a third kind of device, the biological research in canisters, BRICs, which have been used by NASA for the last 25 years for a variety of microbiological and plant studies. They allow you to, oops, wrong button. They allow you to inject fixative or inject nutrients, or do things that the astronauts can do during an experiment. We also use free flyers. If you have a microbial payload and you have some specific questions that can be answered without human intervention, these CubeSats are a standard way of putting together, and you can do chromatography. You can do a lot of stuff in a 5-inch cube, miniaturizing technology, and those are available technologies. I've thrown in, and this may seem like a hodgepodge, but a couple of examples of funded investigators in this area. Our colleague, Dr. Venkateswaran, whom I'm sure some of you know at JPL, has been studying under the aegis of a different segment of NASA, the Space Mission Director, the Science Mission Directorate, which sends out probes to other planets and other bodies. Venkat has been very interested and has studied for the past many years the clean rooms at JPL to see just how clean they are and what's there after you sterilize. And surprise, surprise, there's lots there after you're done making them clean. Venkat is in the middle of a three-flight sequence where he's sampling, collecting microbes from surfaces, air, and water, subjecting them to 21st century analysis, genomics and other omics. This is of interest to both the fundamental science, which I represent, and the human health and performance. This is meant to be a series of experiments. You can see the sort of problem that they have up there, just like your shower at home. That's where they hang their towels when they're wet. And guess what? There's stuff grows there. And we're studying it and trying to make sure that we understand it and its impact on the crew. This is an example of a study which is just being kicked off. It came from a recent solicitation by Cheryl Nickerson at Arizona State, who some of you may be aware of the fact that she's about 10 or 12 years ago, she started doing experiments looking at virulence of Salmonella, and has uncovered increased virulence during spaceflight, and also has taken steps towards understanding the mechanisms for those. This proposal is one that I alluded to before, and this will look at 30 generations to get a longitudinal study of how a set of microbes adapts over the course of time. Obviously, with full genetic and genomic workup is the plan. A second clarion recommendation from the Decadal survey that the National Academy did for us was that we use 21st century tools. And my boss, Marshall Porterfield, has taken that to heart, and his flagship project he's called Genelab. And that is to try to understand the omics of all of the specimens that we try to study. So this is why Cheryl actually was responding to a solicitation where we said we want a team, we want multi-omics. We don't really care which of the organisms you use. So we're in the midst. We're probably a third of the way through getting this public bioinformatics repository going, which will contain both in itself and also through Federation spaceflight results, so that and the part of the plan is also to develop tools for investigators to be able to go into the database and look across omes or across species or to actually work this and find out what's going on. This is a cute slide. It helps me understand, since I'm not a geneticist, what all the pieces and parts are. I'm sure this is like bringing coals to Newcastle for you guys. You know all this stuff. This systems biology approach is our second response to our decadal survey, and that is to bring space biology into the 21st century by using genetic and genomic tools to understand how organisms are responding. There is the Genelab database has been seeded with legacy data that are in the published literature and that have been published and put in other databases. There's a website on there. You can go in and look at what's there. It's not. There's more in there than 3, but they're having problems with their search engine, so that's all I was able to get on one page. There are about 50 legacy data sets, a couple of which are our experiments that have been flown recently. Just to give you a feel, for the past 10 or 12 years, across the portfolio in human biology and space biology, there have been about 60 or so projects by 38 or 40 PIs looking at a variety of problems, the inventory and surveillance, like I described of VenCat, how microbes adapt to spaceflight, pathogenesis. Cheryl Nickerson and a couple of others have worked in that area. Host-microbe relationships. Plant microbes is something that we haven't done much of and we want to do. And the viral research has been mostly on the human side. Humans shed reactivated herpes virus in space. It's still kind of mysterious about why that's going on. But that's been an active set of investigations. Here are some websites: our division, the Space Biology, Human Research, the Genelab Strategic Plan, and a task book, which is a compendium, a searchable database of all the tasks that have been funded in space life sciences since 1994. So you can go in there and look to your heart's content and see what's there. And that's really what I had to say. I'd like to hear something from this committee about microbial evolution over time and the particular challenges. We're interested in radiation and weightlessness and the stress of being contained. But I'm sure there are stressors in buildings and stressors in other structures and in submarines that cause microbes to perhaps behave in a different way than you might expect them to otherwise. And we need to understand that. NASA needs to understand how what goes on in this area, because if you're going to send a spaceship to Mars, which eventually we will, or even consider the ISS. I liken the ISS to a cruise ship. We can't bring it into dock, take all the people off, clean it, and set it off again if something goes wrong. And if something goes wrong on the way to Mars, I mean, we need to be able to predict how the microbial population is going to behave and how to control it, and that needs to be factored into the building of spacecraft. So that's all. Thank you.
Thank you. Very interesting.